Спасибо. Good morning, dear students. Today we are going to continue our discussion. Today's lecture is devoted also to physiology of blood, and we are going to discuss following questions: white blood cells or leukocytes and hemostasis. Let me remind you that blood is a type of high specialized viscous fluid connective tissue that circulates through the circulatory system of our body. All functions of blood are related to transport of different substances, and according the substance transported, according the body needs, ничего, просто ничего, просто according the substance transported and according the body needs, all functions of blood are subdivided into respiratory, nutrient, excretory, regulatory, and defensive functions. As you remember, a volume of blood plasma stays around 45%, and volume of blood cells, blood-formed elements suspended in blood plasma, stays around 45% of the total circulating volume. Here on that slide, you may see this ratio in between blood-formed elements, mainly red blood cells, because they are much more numerous than the rest one, and blood plasma. This percentage of blood-formed elements to the blood plasma called hematocrit, as you remember. Here on that slide, you also may see the blood composition. As you remember, in average 70 kilogram men, the volume of blood circulating stays around 6 to 8% of the body weight. Blood composed of a liquid portion called blood plasma and blood-formed elements. The blood-formed elements are red blood cells or erythrocytes. We have discussed their functions at previous lecture. White blood cells or leukocytes and platelets or thrombocytes. Today our lecture is devoted to the functions of white blood cells and thrombocytes. White blood cells or thrombocytes. The leukocytes are subdivided into two types according their granules which are present in their cytoplasm. There are granulocytes, which also are subdivided into neutrophils, eosinophils, and basophils, and agranulocytes, monocytes, and lymphocytes. The granulocytes are so named because they have granules in the cytoplasm that contain biological active substances involved in inflammatory and allergic reactions. Young granulocytes have horseshoe-shaped nuclei that become multilobed as the cells grow older. Neutrophils. The neutrophils contain neutrophilic granules in their cytoplasm that are smaller and paler than the other types of leukocytes. They do stain with an acidic or basic dye. Nuclei of these leukocytes are lobulated with thin bridges between the lobules. The granules in cytoplasm contain various proteolytic enzymes, as for example, catalase, myeloperoxidase, elastase, defensin antimicrobial protein, and variety of other proteases that help them to destroy invading organisms. They they have a short half-life in the circulation, only 4 to 6 hours. Then they enter the tissues by a process called diapedesis and will live there 5 to 6 days. Enter GIT, gastrointestinal tract, and then lost from the body. Invasion of the body by bacteria triggers the inflammatory response, and ingestion of bacteria by endocytosis. As you remember, it is called phagocytosis. So the neutrophils are phagocytes. Functions of neutrophils are phagocytosis. Each neutrophil is capable to engulf to eat 5 to 20 microbes. They also participate in inflammatory reactions of an organism, participate in inflammatory response due to the variety of substances they contain in their cytoplasmic granules. Next type of leukocytes are eosinophils. The eosinophils, as you see here, have bilobed nuclei. Their granules are large and pronounced. They are stained red with the acidic dye called eosin. They release proteins, cytokines, chemokines that produce inflammation, but also are capable to kill invading organisms. Functions of eosinophils are following. Eosinophils are weak phagocytes. They are involved in inflammatory and allergic response. They also have defense function, so they promote defense against parasitic roundworms, especially abundant in the mucosa of gastrointestinal tract, respiratory and urinary tracts. They also produce precursor of a plasmin called plasminogen, which participate in hemostasis. Basophils. Basophils are distinguished from the other granulocytes by presence of dark basophilic granules in cytoplasm, stained blue with a basic dye. Their nuclei are large and varied in shape. They release proteins and cytokines. They contain histamine and heparin release them, increasing regional blood flow, facilitating the transport of other leukocytes to area of infection. As you know, the histamine acts as vasodilator and heparin prevents a blood coagulation, anticoagulant. They also essential for immediate type hypersensitivity reactions. So, functions of basophils are to participate in inflammatory and allergic response and due to release various substances from their cytoplasmic granules. All granulocytes have cytoplasmic granules that contain biological active substances involved in inflammatory and allergic response. All of them act as phagocytes. Basically, all leukocytes act as phagocytes. Next type of leukocytes are monocytes. The monocytes have abundant cytoplasm and a darkly stained kidney or U-shaped nucleus. They enter the tissues and become tissue macrophages. This time, macrophages include the Kupffer. These tissues, tissue macrophages, so-called tissue macrophages, include the Kupffer cells in the liver, pulmonary alveolar macrophages, microglia in the brain. In the past, by the way, they have been called reticular endothelial system, and nowadays it is called tissue macrophage system. Functions of monocytes. The monocytes are much more powerful phagocytes than the other leukocytes, much more powerful than neutrophils. They are capable, one monocyte is capable to engulf around 100 bacteria. Monocytes also involved into the immune response. They recognize antigen and are involved into antibacterial, antiviral, antiparasitic, anti-tumor immunity due to release of various substances. More than 100 substances are secreted by monocytes, as for example, interleukins, interferon complement system proteins, cytokines, clot promoting factors, prostaglandins, activators of plasminogen, etc. And the last type of leukocytes, which are called lymphocytes. Lymphocytes. Here on that slide, you may see how they look like. Some lymphocytes are formed in the bone marrow, but most are formed in the lymph nodes and spleen from precursor cells that came from the bone marrow. Lymphocytes enter the circulation via the lymphatics. Only 2% of the body lymphocytes are found in the peripheral blood. Most of the rest are found in the lymphoid organs. Their functions in the lymphoid nodes or spleen is to defend against foreign microbes in the reactions of immune response. As you know, there are two types of lymphocytes, so-called T lymphocytes and B lymphocytes. B lymphocytes are involved in humoral mediated immunity and are subdivided into these subtypes: cytotoxic T cells, helper T cells, memory T cells, and suppressor T cells. And B lymphocytes are involved in the humoral mediated immunity. Subtypes of T cells have been identified using fluorescent monoclonal antibodies to the specific cell surface antigens known as CD antigens. As I told you, there are four major types of T cells, which are involved in cell-mediated immunity: cytotoxic, helper, memory, and suppressor T cells, and two types of B cells that are involved in humoral mediated immunity. I will tell you briefly about the function of each subtype of T and B lymphocytes. So the B lymphocytes, which are involved in the humoral immunity, protection by antibodies, and major defense against bacteria, will mature in the bone marrow. Their precursors mature in the bone marrow and then differentiate into memory B cells, which are responsible for accelerated response to a second exposure of the same antigen. And they also differentiate into the colony or clone of plasma cells, which secrete immunoglobulins or antibodies. As you remember, there are 5 classes of immunoglobulins: A, G, M, D, and E, found mainly in gamma globulins. So memory B cells and plasma cells. As I told already, cell-mediated immunity protection is by T lymphocytes, which react directly with foreign cells, with tissue transplants, cells infected by organism, cancer cells, and the bone marrow stem cells, which migrate to the thymus. The thymus transforms them into T lymphocytes, which will differentiate into helper T cells or T4 cells, process the antigen for interaction with B cells. Memory cells, which are responsible for accelerated response to a second exposure to the same antigen. Suppressor T cells, which decrease immune response of T and B cells. And cytotoxic T cells, or so-called killer cells, or T8 cells, which destroy transplanted and other foreign cells by cell rupture due to ion uptake or insertion of toxin into the cell. The normal percentage of white blood cells ranges for the various leukocytes are as follows: neutrophils 50 to 70%, lymphocytes 20 to 40%, monocytes 3 to 12, Eosinophils 0.5 to 5, and Basophils 0 to 1. A white blood cells count that determines the relative percentage of each type of leukocytes, called a differential white blood cells count, or in Russian interpretation it is called leukocytic formula. Deviation from the above percentage may indicate serious pathological conditions. For example, high neutrophil count or so-called neutrophilia, when it increases above 70%, often signal localized infections such as appendicitis or abscesses in some part of the body. Also, it may increase in exercises, in stress, and corticosteroid drug therapy. Neutropenia, which is when neutrophils count reduces below this range, below 50%, is observed in conditions in which there is a marked decrease in the number of neutrophils, for example, in fever or influenza. Eosinophilia, as you understand, is high eosinophil count, may indicate allergic reactions or invasion by parasitic worms. Lymphocytosis is present in whooping cough and some viral infections. So, differential white blood cells count is the relative percentage of each type of leukocytes. The total white blood cells count normally fluctuates in this range, 4 to 9 times 10 power 9 per liter, or 4 to 9,000 in cubic microliter of blood. When white blood cells count increases above this range, above 9, it is called leukocytosis. When it reduces below that range, it is called leukopenia. The leukocytosis are of two types: physiological leukocytosis and pathological leukocytosis. The physiological leukocytosis will occur due to redistribution of blood between different organs depending on their activity. The variants of physiological leukocytosis are following: alimentary leukocytosis, which happens after the meal, after the food intake; the myogenic leukocytosis after physical exercises, after intensive muscular work, or followed physical stress, so-called physical stress, and emotional, following emotional excitement, following emotional stress, it is called emotional leukocytosis. Also, it happens in normal physiological pregnancy. Pathological leukocytosis. The pathological leukocytosis will occur in the case of activation of the process of leukopoiesis in inflammation or infection diseases and allergic response, and leukemia or blood cancer. The leukopenia is pathological, and leukopenia related to reduction of leukopoiesis. It also related to excessive white blood cells destruction, and it is followed by chemotherapy, radiation therapy, and in X-ray disease. Types of immunity. As we told already, the white blood cells participate in the immune response of an organism. The immune system, as you know, protects our body from invading microorganisms. The immunity can be divided into non-specific or innate or natural immunity, and specific or acquired immunity. Natural immunity provides the basic means for the destruction of organism. It provides like the first line of defense against infections, but also triggers the slower but specific acquired immune response. Simply speaking, it is action against any foreign or abnormal cells. This mechanism includes physical barriers to infection, skin and its secretion, which have an acid pH, mucus in the respiratory system, in the GIT, in the digestive tract, acids and enzymes or digestive juices, as you can see here, sweat and nasal secretion, the growth of non-pathogenic bacteria in the gastrointestinal and urogenital tract. It also includes inflammation, complement activation, and natural killer on large cytotoxic cells activity, means phagocytosis. When invasion of the tissue occurs, interferons alpha and beta produced by the virus-infected cells protect host from the viral infection and stimulate natural killer cells. Interferons alpha and beta produced by virus-infected cells inhibit viral replication and stimulate phagocytosis, activate natural killer cells, enhance immune response. The complement system is a system of plasma enzymes identified by the numerals from C1 to C9. They enhance immune, allergic, and inflammatory reactions. They attract phagocytes to the site of injury, make holes in membrane of microorganisms, causing them to rupture. The natural killer cells are large granular lymphocytes which kill virus infected cells and some types of cancer cells. Inflammation, a response to damaged tissue characterized by reduced by pain, by heat and swelling, and they will be destroyed and tissue repaired. Interleukins are hormone substances produced by the lymphocyte. There are 5 types of them. And then they will stimulate clones of B and T cells, cause plasma cells to secrete immunoglobulins. Acquired immunity or specific immunity is the ability of leukocytes, lymphocytes to produce antibodies that are specific for one of the many millions of foreign agents that may invade the body. The acquired immunity has 2 components. Humoral immunity, which is mediated by B lymphocytes, and cellular immunity, which is mediated by T lymphocytes. The humoral immunity, which is mediated by circulating immunoglobulins, antibodies in the gamma globulin fraction of the plasma proteins. Immunoglobulins are produced by B lymphocytes and they activate the complement system and attack antigens. Humoral immunity is a major defense against bacterial infections. Cellular immunity is mediated by T lymphocytes. It is responsible for delayed allergic reactions and rejection of transplants of foreign tissues. Cellular immunity constitutes a major defense against infections due to viruses, fungi, and bacteria, and it also helps defend against tumors. Now let's discuss some aspects of hemostasis. The process of hemostasis mostly depends upon the function of blood-formed elements called platelets or thrombocytes. The platelets or plaques are blood-borne cell fragments which contain organelles and enzymes, but no nuclei material. They bud off from megakaryocytes and giant multinucleate bone marrow cells derived from the myeloid stem cell line. The normal platelets count in blood is ranges in between 150 to 380,000 per cubic microliter of blood, or 150 to 380 times 10 power 9 per liter. The role of platelets is to help prevent or arrest bleeding in a process called hemostasis. The number of platelets in peripheral blood increases in the daytime and decreases at night. Platelets survive from 2 to 5 days, average 4 days. The lifespan is about 4 days. platelets are rapidly destroyed in bleeding, and factors that take part in coagulation, which are so-called platelet clotting factors, are released, are liberated into the blood plasma. They also liberate substances which will cause vasoconstriction, as for example serotonin, epinephrine, norepinephrine. Quantitative deficiency of platelets leads to thrombocytopenia. It results in abnormalities of hemostasis, such as prolonging of bleeding time, qualitative deficiency of thrombocytes or thrombocytopathy caused by insufficiency of platelet factors. So there are two variants of fluctuation, platelets number, quantitative deficiency thrombocytopenia, and qualitative deficiency thrombocytopathy. Hemostasis. The hemostasis is the process of forming clots in the walls of damaged blood vessels and preventing blood loss while maintaining blood in a fluid state within the vascular system. A collection of complex interrelated systemic mechanisms will operate to maintain this balance between coagulation and anticoagulation. When blood vessels are ruptured, three mechanisms will arrest bleeding, or three mechanisms of hemostasis are initiated. The first mechanism is a constriction of blood vessels or vasoconstriction, which is a reflex vascular spasm of smooth muscle in the walls of the blood vessels. Then, second one is a platelets plaque formation of platelets plaque adhere to exposed collagen, release growth factors to increase endothelial smooth muscles and fibroblast cells, and release serotonin, the thromboxane A2, to constrict blood vessels. And the third stage, the third mechanism or third stage is a blood coagulation or clotting, involving enzymes and chemicals called clotting factors, which ultimately result in thrombin catalyzing the formation of fibrin. So there are three mechanisms which may arrest bleeding, or three mechanisms of hemostasis: vascular spasm or reflex vasoconstriction, the platelet plaque formation, and blood coagulation or clotting. Vascular spasm. Damage to the wall of the blood vessel leads to a rapid, here it is, rapid, smooth muscle contraction. This may help to narrow the vascular defect and reduce blood flow. Vascular spasm alone, however, cannot arrest blood loss. Platelets normally circulate freely, but when they are exposed to tissue collagen following vascular damage, they become adherent or sticking to the vessel wall and to each other. At the same time, they release a number of chemicals, which act as local humoral factors, as for example, serotonin, adrenaline, thromboxane A, ADP, and this will increase platelet thickness, resulting in positive feedback favoring the formation of a ever large platelet plaque, which can stop bleeding by occluding the opening. The bleeding time, the time taken for visible blood loss from a small puncture wound to stop, is usually less than 5 minutes. But this may increase considerably if platelet function is defective. The platelet adherence to undamaged blood vessels is normally inhibited by prostacyclin released from from endothelium, which decreases platelet thickness. Here you may see the how this process takes place. The von Willebrand factor will initiate the process of released from the tissues, damaged tissues, will help the thrombocytes to stick to the collagen fibers exposed. The same time, thrombocytes release a number of chemicals called platelet clotting factors, and these platelet clotting factors will influence on the process of adhesion of platelets to the formation of the platelet plaque. As you see, the small amounts of thrombin, which is formed already simultaneously to the vascular thrombocytic hemostasis in the process of blood coagulation by extrinsic system, will convert the temporary reversible platelet plaque, so-called temporary reversible platelet plaque, into irreversible platelet plaque, and thrombostatin, one of the platelet clotting factors, will cause retraction of the platelet plaque, and the result of adherence, aggregation of platelets, and retraction of platelet plaque is the formation of temporary hemostatic plaque, or platelet plaque. These are phases of vascular thrombocytic hemostasis. vascular spasm, the constriction of the vascular wall due to epinephrine, norepinephrine, and serotonin, reflex constriction. Simultaneously to that, formation of platelet plaque initiated due to adherence and aggregation reversible, irreversible due to thrombin, and finally retraction of platelet plaque due to thrombostenin. Result of this is a temporary hemostatic plaque formation or platelet plaque formation. Here on that picture you also may see this process, the process of platelet plaque formation. Next stage of hemostasis, as you remember, called coagulation of blood or clotting. The clotting or coagulation depends on a family of plasma proteins, as you see here, called the plasma clotting factors. The Roman numerals, these Roman numerals are clotting factors, plasma clotting factors. Activation of clotting factors is indicated by A. These plasma clotting factors are a family of plasma proteins. Several of them are manufactured in the liver in vitamin K dependent reaction. Normally, they are present as inactive proenzymes, but once activated, each factor activates the next by proteolytic cleavage. As I told already, synthesis of some of them, for example, factor 2, factor 7, factor 9, and factor 10, requires vitamin K. Factor 8 is absent in hemophilia, called antihemophilic factor. Blood does not normally clot within healthy blood vessels. A thrombus is a clot within a blood vessel. Antithrombin 3 inhibits thrombin and factor A activated and is potentiated by heparin, a powerful anticoagulant. Clotting can be activated through two different mechanisms, referred to as the extrinsic mechanism and intrinsic mechanism. Also, these mechanisms called pathways, extrinsic pathway and intrinsic pathway. The activation of intrinsic pathway or mechanism in vitro occurs when blood contacts with glass, in vivo when blood is exposed to the collagen fibers underlying the endothelium in the blood vessels. The time of activation of this mechanism fluctuates from 5 to 7 to 10 minutes. It includes activation of 12, 11, and 9 factors. Then the complex of 8 and 9 activated will activate factor 10. The extrinsic system is triggered by the release of tissue thromboplastin, its interaction with the factor 5 and 7 in the presence of calcium ions, and results of this activation is factor 10 activated, which is called tissue prothrombinase, tissue prothrombinase. By the way, this will provide the thrombin formed by this mechanism, extrinsic pathway, will provide aggregation of platelets, as I told you. So here you may see how they will interact. The extrinsic pathway, which is fast mechanism, 10 to 15 seconds it takes, and intrinsic, which is slow, 5 to 10 to 7 to 10 minutes. This coagulation cascade of coagulation finally results in the conversion of prothrombin, which is also plasma clotting factor 2, to activate thrombin. Thrombin catalyzes first the conversion of fibrinogen factor 1 to fibrin monomers, and then their polymerization into cross-linked fibrin strands, fibrin polymer. The resulting clot, which traps red blood cells and other blood-formed elements, bridges the opening in the blood vessel wall, as you may see here. After some time, clot retraction pulls the damaged edges of the blood vessel together, squeezing out the clear serum. Here on this slide, you also may see one more variant of a scheme that presents blood clotting mechanisms, blood coagulation mechanisms. The blue color will show the coagulatory cascade, which finally called extrinsic mechanism or extrinsic system, which finally results in so-called tissue prothrombinase, the factor 10 activated. And the yellow one will show the intrinsic mechanism or intrinsic pathway. It is also coagulatory cascade, proteolytic cascade, which results in also in the inactivation of factor 10. Then factor 10 activated will initiate the conversion of plasma clotting factor prothrombin to thrombin, and which in turn, by the way, it needs 2 to 5 seconds. And then 10 to 15 seconds needs to convert fibrinogen to fibrin. Then fibrin polymerization takes place fibrin soluble becomes fibrin insoluble, and definitive hemostatic plaque or fibrin clot finally is formed. Here you may see the electron micrograph of erythrocytes trapped in fibrin mesh in the process of fibrin clot formation. Abnormalities of hemostasis. In addition to clotting abnormalities due to platelet disorders, deficiencies of clotting factors, can lead to severe bleeding problems. This may arise because of liver failure or vitamin K deficiency. Both will inhibit clotting factors synthesis, because I told you that synthesis of some of them requires vitamin K, so manufactured in vitamin K dependent reactions. It can be also inherited deficiency of any clotting factors. The most common and most famous is hemophilia, factor 8 I have shown you earlier, and also congenital deficiency of von Willebrand factor. The tendency of blood to clot is balanced in vivo by limiting reaction that tend to prevent clotting inside the blood vessels and to break down any clots they do form. The lysis of blood clot inside the blood vessel is called fibrinolysis. This depends on the proteolytic enzyme plasmin, which is formed from the inactive plasma protein called plasminogen, following activation of the coagulatory cascade. Plasmin catalyzes the breakdown of fibrin, a process called fibrinolysis, and then phagocytic cells remove the clot debris. A number of natural factors act as anticoagulants, inhibiting clot formation in normal vessels. Some are used to prevent clotting outside the body in vitro. Few can be used both in vivo and in vitro. The various anticoagulants are as follows. Those which activate antithrombin, heparin, coumarin derivatives, for example, warfarin, oxalate, citrates, antithrombin, and anticoagulants can be classified as primary anticoagulants and secondary anticoagulants. The factors that accelerate hemocoagulation are increased body temperature, presence of calcium ions, unsmooth surface blood vessels walls, mechanical distortion, etc. Factors that inhibit hemocoagulation: decreased body temperature, citrates, oxalates, which can bind with calcium, heparin and antithrombin, which binds thrombin, smooth surface of blood vessels. Both sympathetic and parasympathetic nervous system increased tone will cause hypercoagulation. Hypercoagulation always takes place following hypercoagulation. It is like secondary and caused by deficiency of procoagulants or activation of anticoagulants. So that's all what I wanted to tell you about the physiology of blood. We have discussed the composition of blood, physiological, physicochemical properties of blood. We have discussed the blood-formed elements and their functions. And we also have discussed the process of blood, the summary of events involved in the hemostasis. Thank you for your attention. Goodbye. The lecture is over.